Hello, my name is Leslie Mamani, and I'm a student from San Marcos University. I study at Pharmacy and Biochemistry faculty, and here I present the dynamic modeling of renin-angiotensin system perturbation by SARS-CoV-2 and the proposed drug for COVID-19 treatment. As an introduction, we need to note that the RAS system is crucial for blood pressure and the electrolyte balance regulation, and it is composed by different peptides and enzymes, but here we have the main ones: ACE1, ACE2, and MAS. ACE1 has the function of converting angiotensin 1 into angiotensin 2. ACE2, apart from its physiological function, it acts as a SARS-CoV-2 receptor, and the MAS receptor, it can interact with angiotensin 1,7. What about 20E? Why it is here? It also can interact and activates MAS. Having this in mind, here we have the methods. First, we adopt a code in Mathematica that we obtained from the study made by Fabrizio Buzzi, and we adapted to Python. Then we introduced the interaction values into the mathematical model, and they were related to the SARS-CoV-2 infection and the 20-hydroxyedison that we mentioned before as 20E. About the infection, we use the CT, which is a function inversely related to the viral load, and 20E, we use the maximum concentration after a specific oral dose and the half-time life. Then we did the mathematical model modification. The model was composed by 9 ordinary differential equations, and we specifically modified the mass-active differential equation. Then we did the simulation without and including the virus and the treatment. And for this, we used Python and Jupyter. And also the Python libraries used were NumPy, SciPy, and Matplotlib. Finally, to process the results, we used the formula that we can see here in the slide. In order to understand better the results, here we have the RAS system model that we use to this investigation. Here we can see the different peptides involved here, and also the enzymes that are expressed as reaction rates. In these pictures, we see that the variation of concentration of the components of RAS system during 300 seconds. In the first one, we can see the angiotensin 1,7 that it has a higher concentration than the other components, but then with severe infection, so when the virus is introduced, the angiotensin 2 concentration increases several points compared to the other components. But then, when 20E is introduced, the concentration of mass active increases too. Here in this table, we can see in a better way the results that we obtain. As a summary, we can see that SARS-CoV-2, we know the virus, disrupts in all downstream components of RAS system, but specifically on these ones. But as we can see, when 20E is introduced, it can reduce the perturbative effect of the virus on specifically mass active. Why it is important? Because mass active or mass activation, in fact, it's related to a specific axis that is the ACE2 angiotensin 1,7 mass axis, and it has different beneficial effects related to vasodilation, anti-inflammatory, antithrombosis, and other actions. But on the other hand, angiotensin 2 and its interaction with AT1R is related to all those negative effects that we have as a result from the disease. So in conclusion, this present study developed a model that allows us to evaluate the effects of the virus and the 20E treatment on RAS system. And it was concluded that the virus exhibited a strong perturbative effect, but it can be reduced by 20E through mass activation. Moreover, our model constitutes a tool for testing hypotheses about the effects of drugs in disease-perturbed systems as RAS. So the possibilities that we have are a lot, and it's so fascinating to think about that. Thank you.